Hello, my name is Hannah Zima, and I'm a second-year grad student at the University of Arkansas in the Department of Entomology and Plant Pathology. And today I'll be going over my thesis efforts, the diseases of Cannabis sativa and the epidemiology of Neofusococcum parvum. Before I go into my thesis efforts, I thought it would be best to quickly go over hemp in the U.S. And as of 2018, the passing of the Farm Bill allowed for the cultivation of hemp for both research and commercial interests. It is a dioecious plant, meaning that male and female parts are on individual plants, and this picture here is just to show you what a male plant looks like versus a female. It is photoperiod sensitive, meaning that it needs to have specific light conditions in order to flower, and there are 3 species of cannabis: Cannabis sativa, Cannabis indica, and Cannabis ruderalis. And all you really need to know about ruderalis is that it's photoperiod neutral, so it actually doesn't really need specific light conditions in order to flower. And the legal definition for hemp is that it has to have less than or equal to 0.3% THC, whereas marijuana has more than 0.3% THC. And this picture here is to show you how the CBD compound versus the THC compound look very similar. So for chapter 1 of my thesis, it is a survey of cannabis diseases in Arkansas and California. And the justification is that it's a new crop in the U.S. and disease surveys haven't really been done for this crop. And here's me with some growers in Arkansas and some growers in California. And this down here is just to give you an overview of the scope of production that I was seeing. So for chapter 2 of my thesis, it is the epidemiology of terminal dieback of hemp, or Neofusococcum parvum. And again, the justification for this chapter is that this is a novel disease on hemp in the U.S. The first report was in Arkansas by my lab specifically. And what we've been doing is we've been establishing a culture collection of Neofusococcum from hemp and non-hemp hosts, and we then have been investigating various pathogenicity assays, whether it's a whole plant or detached leaf. And as you can see here, I've done, in the various hosts I've been investigating have been blueberry, grape, blackberry, and oak, all that have been reported to having N. parvum on them. And to round out the last of my thesis efforts, it is an analysis of the whole genome of N. parvum. And what I've done is I've taken some reference isolates from my culture collection, and I used multiple primers to make sure that it was N. parvum. And I then sent them to Novogene, which is a sequencing company, and they sequenced my isolates for me and sent back huge files, large data files that I've been slowly and painfully trying to go through, either with the university's supercomputer or with the help of professors in my department. And here is just some phylogenetic tree that I have made with some people in my lab, just again showing you how my isolate from hemp compares to other isolates from grape and other isolates that are from Botrytis viriaceae. All right, thanks guys.